In this presentation, I'd like to walk you through the HiChIP MNase workflow so you have a better understanding of what is involved in running the assay in the lab. The HiChIP MNase workflow can be completed in 2.5 days, and that is going from sample to a sequencing-ready library. It consists of 6 stages and a total of 18 steps. Day 1 is for cross-linking, digestion, cell lysis to extract the chromatin fragments, and incubation of the chromatin with the antibody of interest overnight. So that would be stage 1, 2, and the first part of stage 3. Day 2 is for immunoprecipitation and proximity ligation. So that would be the second part of stage 3 and stage 4. At the end of stage 4, you would have generated proximity ligated DNA that was associated with an immunoprecipitated protein of interest. This DNA is then converted into a sequencing library on day 3, which includes stage 5 and 6. There are several safe stops throughout the protocol, and there are also QC steps built into the workflow. Now, this is an overview of the workflow. In the next slides, I'll walk you through the workflow stage by stage, and I'll share with you our tips and best practices. Starting with stage 1. In stage 1, the chromatin is fixed in the nucleus, then digested with MNase. Following digestion, the cells are lysed to extract the chromatin fragments. For cross-linking, we use DSG and formaldehyde. So formaldehyde cross-links protein to DNA and proteins to proteins that are in close proximity. DSG has a longer spacer arm. It's a strong protein-to-protein cross-linker, and it cross-links proteins to proteins that are more distant. So when you use DSG in conjunction with formaldehyde, it increases the long-range information captured in the library. When cross-linking, we rotate the cells in a standard tube rotator so they don't settle. The HiChIP MNase assay does not require sonication. It will only rely on the enzymatic fragmentation or the digestion with MNase. So it's important to achieve a good digestion in order to generate a high-quality library. For optimal digestion, we recommend to freeze the cells before cross-linking. The number of cells you'll start the assay with depends on your protein of interest, but that number will influence the amount of MNase you're using to optimally digest the chromatin. So to help with an accurate cell count and to reduce cell clumping, we recommend to use a low-speed spin and a swinging bucket rotor when you harvest the cells, and we recommend to count before you freeze the cells. So you would harvest the cells at low-speed spin, wash with PBS, count, aliquot the number of cells you're going to start the assay with, spin down, discard the supernatant, and then freeze the cell pellet by placing it at -80. Then you can proceed with cross-linking and digestion. We provide sample preparation guidelines for 1 million, 5 million, and 10 million cells. Moving on to stage 2. This stage is a QC step that is included early on in the protocol that will allow you to confirm that the chromatin is properly digested. It will also allow you to quantify the yield of clarified lysate or chromatin that you recovered from the cell sample. So how do you do that? To confirm that the chromatin is properly digested, you will take an aliquot from the total lysate you recovered after cell lysis at the end of stage 1. If you're going to proceed with the rest of the protocol on the same day, you can place this lysate in your ice bucket. Otherwise, we recommend to store this lysate at -80 for later. So this aliquot is essentially cross-linked chromatin. You will first start by reversing the cross-links, degrading the proteins with proteinase K, then purifying the DNA. You will use this purified DNA to run on an analysis instrument. You can use either tape station, bioanalyzer, or fragment analyzer. You'll also quantify this purified DNA with a qubit fluorometer that will enable you to determine how much lysate you recovered from your cell sample. Here is an example of a digestion profile analyzed with a tape station. That first peak represents mono-nucleosomes. Here are the dinucleosomes, and here are the trinucleosomes. The profile we're looking for has 40 to 70% mononucleosomes. In order to determine the percentage of mononucleosome, you're going to create a region that includes that first peak, which represents the mononucleosomes. So on a tape station, this first region is between 50 to 250 base pair. This is where you see the first peak. It may vary with different systems, but you're always looking at that first peak. When you create this region, a region table will be automatically generated, and it outputs percent of total. So how much of your DNA is within 50 to 250 base pair? This is the percentage of mononucleosome, which is in this case 52.22%. Here is an example of a digestion profile analyzed with a fragment analyzer. Again, here are the mononucleosomes, dinucleosomes, trinucleosomes, and based on the smear analysis, the mononucleosomes account for 48.4%, meaning these two profiles pass QC. Now, a few tips that I would like to share with you here. It's important to load that tape to spec, so avoid overloading the tape. Also, we recommend for a specific system the tape to use. For example, for tape station, we recommend to use the HSD5000 tape and not the D1000. And that's important because it gives you a good resolution for the size that we're actually looking at for the digestion profile. Now, if your chromatin is not optimally digested, so if the chromatin is underdigested with less than 40% mononucleosomes, or your chromatin is overdigested with the mononucleosomes accounting for more than 70%, we provide guidelines for how to adjust this digestion. Now that you've determined that the chromatin is properly digested and you quantified the lysate, you can proceed to stage 3. You will start by incubating the chromatin or your clarified lysate with the antibody of interest overnight. Now, the question is how much antibody and how much chromatin to use, which is also a question that's asked in ChIP-seq. To address this question, we validated a list of antibodies. This list includes CTCF, histone marks, and transcription factors. So what we did is we tested antibodies from different suppliers to make sure that the antibody recognizes the epitope under the assay conditions. We also ran different titrations. So for each antibody, we provide guidelines for which supplier we recommend and what is the amount of chromatin and the amount of antibody that we recommend to use for the HiChIP assay. I've highlighted CTCF, trimethyl H3K4, and acetyl H3K27 because we also offer a HiChIP library preparation with these antibodies in-house as a service. Now, after incubation, you're going to pull down on the antibody protein DNA complex with protein AG beads. Now that you have the chromatin fragments bound to the beads, you're going to run the proximity ligation reactions, starting with end polishing, where you're going to repair the ends of the chromatin fragments. You will then add a custom-made biotin-tagged bridge or adapter to those repaired ends. Then the fragments that were co-localized in the 3D space are going to be ligated to each other through the bridge. And here is where you're going to generate that proximity ligated chimeric DNA molecules. After ligation, you're going to reverse the crosslinks, degrade the proteins, and purify the DNA with a bead cleanup. For the intra-aggregate ligation and crosslink reversal reactions, they are incubated for 1 hour, but they can also be incubated overnight for convenience. All of these reactions are carried out in a thermal mixer with shaking. I'm just showing you an example of a standard thermal mixer that we use in-house that has a heating and cooling function. So there isn't really any specialized equipment that you need to run the assay. So now that you've generated the proximity ligated DNA, you're going to convert it into sequenceable molecules in the last 2 stages of the assay. Stage 5 is library preparation. As I mentioned, there is no sonication step prior to library preparation, so you will directly proceed to library preparation. These steps are standard for any NGS library prep. So you'll first repair the ends, add the adapter, and purify the adapter ligated DNA. Then before PCR amplification, we include a streptavidin bead pull-down step, which will be pulling down and enriching specifically for the proximity ligated biotinylated DNA molecules before you amplify. So we run the amplification on the beads. And then following amplification, we size select the library with price select beads. And then we run this library on a fragment analyzer, bioanalyzer, or tape station to check that it has the expected size range. So we expect to see the library between 350 and 1,000 base pair, and we expect to see that laddering in the final library, which is reflective of the laddering we see with the MNase digestion. So this library is now ready for paired-end sequencing on any Illumina sequencing platform. One thing I would like to add is that the protocol as written is specific for the Dovetail library prep module and primer set that are offered as add-on modules that makes the protocol more streamlined. However, if you prefer to use a library preparation kit that you routinely use in the lab, please reach out to us and we can let you know if that kit is compatible with our assay and how you can integrate it in the workflow. Now that you have the library, we recommend to sequence it to 20 million read pairs in order to run a QC analysis post-sequencing.